My name is Ganasri Aleti, but I just go by Gana. I am an undergraduate researcher in Dr. Anand Chandrasekhar's lab in the Biological Sciences Department, but the program that I'm doing my research through and I'm funded through is the MARC IMSD Express program. Well, we research brain development whenever, obviously brain development in development. And specifically neuron migration during development, trying to figure out what neurons get where, how do they get there, what do they need to get there, what kind of signals do you need. And that's our general lab, but my project is actually working on a piece of tech almost. It's a procedure that we can use to kill off specific cells early on in development so that we can see what happens later. And because if you get rid of something and then later you look and you're like, huh, this fish can't feed properly, it's not moving its jaw. And you're like, oh, what did we get rid of? Oh, the branchiomotor neuron. Cool, so that means that it is important for jaw movement. So things like that, targeted cell killing is very, very useful for that, and that's what I'm working on, specifically in zebrafish. They are really, really useful because number one, they're small. They're small with short generation times, which means that we don't have to wait for 20 years in order to be able to look at one of their brains. You can just overnight, I can have a bunch of embryos that I can work with. Over the course of the next week, in a week, I can have a bunch of larvae that are nearly adult zebrafish come to be able to work with. So the fact that they're so short, it also means that they're easy to manipulate. And by that I mean manipulation of their genome, be able to change up some of their circuitry, and then that way you can pretty easily work with them. So the fact that they're simple, small, very short generation times, it makes them so much easier to observe. Plus, zebrafish embryos are pretty translucent. You can see straight through them whenever you have a microscope. So if you use transmitted light, which is light coming from underneath, you can see pretty much all of it, which makes it really, really useful to use. So it's really important because it'll help with different biology and especially brain studies, because there's still so many things about the brain that we don't know, right? We are basically studying ourselves and we don't know a big chunk of ourselves because we don't know so much about neurobiology and the brain itself. So to be able to have something that works on a really good organism for studies like that, which is a zebrafish, it'll make it really, really useful to be able to do other studies and better understand ourselves through that too. I got involved in this lab in a very weird, kind of roundabout way. I actually met my faculty mentor, my PI, at a volunteer event. And I like to make the joke that we clicked on the level of brown, because both of us are Indian, so we were talking about classical singing, because I'm a classical singer, food, and all of those different things. And afterwards I actually looked up his research and stuff because I was very interested in developmental neurobiology, which is what this lab works in. And then over time just bonded more with him and was like, can I join your lab? And he goes, yeah, let's see how your first semester of undergrad goes first. I want to make sure that you have a strong foundation. And then afterwards I joined this lab. Like I said, I already work in this lab during the school year. And so the thing about working in the school year is that you have so many other obligations. You have classwork, you have part-time jobs. Classwork is probably the biggest part. You have organizations that you have to attend to. I am on the exec board of a couple of orgs on campus. But during the summer is whenever you have all the time in the world. I like to make the joke that I basically sold my soul to the lab for the summer so that I can actually put in the time to make actual strides in my research, because a school year, what I've noticed is that in a semester, in 5 months, I get a lot less done than I do in these 3. along with, making headway into the research itself, because the past 2 years have basically been me preparing to do my experiments, because it's a lot of techniques that I need to know. It's a lot of precision. I can't mess up. I can't go farther than a couple millimeters, or else I risk destroying a couple of embryos. Things like that. Making sure that I can be precise. But other than that, I want to be able to stand in front of people and talk about it. Talk about the research and realize that I actually understand what I'm doing and be able to describe as such. Since last year, I also did summer research here, but it was virtual. So it was mostly just me sitting in front of my computer, talking at my computer while playing a slideshow, and then just recording that and putting it on VoiceThread. I would much rather be able to do something like the Flash Talks and be able to actually stand in front of people and spend that time talking to people and describing my research to them. I think that, I guess the main thing that I really like saying about research is that instant gratification does not appear to be much of a thing. Understanding that small things and how they connect to one another is probably one of the biggest things that you can get out of research, because the understanding that you have to put in a lot and you have to put in work, and it might not seem like you're getting much out of it until way down the road, and you think back and you're like, oh, this is connected to this. I'm helping this project in this way, and this project is actually helpful to 8 other labs for this reason. And then down the line thinking, oh, because of something as small as this, we now have a new piece of technology we can use. We have a new way that we can treat somebody for any sort of ailment that they might have. So I think research itself is definitely long-term gratification. You don't really get much instantly, but it's still rewarding while you're doing it.